This is our last talk this session, and this is Deniz Yorukoglu, who will be speaking about detection and characterization of novel structural alterations in transcribed sequences. Thank you.
Hello, everyone. In this presentation, I'd like to talk about the joint first software with Farashaj. And our paper is mainly about two novel alignment frameworks that we have developed for identifying transcriptional structural alterations, and also an accompanying alignment tool that we have developed called Dissect. And before giving you a general outline of this talk, I would first like to introduce the biological problem at hand and also talk a bit about why we thought it was an interesting problem to formulate computationally. As intermediate transcriptional products take their finalized form, they might lose some intronic sequences within themselves. So basically, the finalized transcript looks more like a combination of exonic sequences. That's why transcriptome to genome aligners like BLAT or GMAP would need to consider the possibility of these long-range gaps on the genome side when aligning transcript to the genome. And they usually utilize the fact that these gaps are spanned by canonical splice signals like GTAG. In our study, though, we wanted to further incorporate some structural alterations to the alignment model. For example, for duplication case, we want to consider the possibility that some certain regions in the genome can get transcribed more than once. And if you want to align this transcript, for example, to the genome, you might be able to align the first two exons properly using a conventional aligner. Yet to be able to align the last exon, you should have an alignment model that can capture such backwards jumps in the genome. A further extension of this would be a rearrangement case in which if you go further backwards in the genome, you might see a rearrangement of the exons rather than a duplication. For the inversion case, we consider the possibility that a certain region in the transcript can be transcribed from a different strand than the rest of the transcript. And even though this example is a double breakpoint example with an internal inversion, it could also be a single breakpoint example with the entire suffix being inverted. For the fusion cases, we consider that we are given two genomic sequences as potential fusion partners. And here we assume that transcription starts in one of these sequences and has a single time jump to the other sequence and continues over that side. The reason we thought this problem was interesting was because there has been recent discoveries upon non-collinear and chimeric transcription events in mice and humans. And also these structural alterations were further confirmed to be enriched in cancer, which might suggest that they are biologically important. And currently available methods for detecting these structural alterations are mostly based on RNA-seq short read mapping. And as next generation sequencing technologies provide us with longer and longer reads, each read is starting to cover more than one exon, in which case the task of short read mapping itself is becoming rather difficult. And the task of de novo assembly would be more easier and more accurate. For that reason, looking at these structural alterations in entire assembled transcripts would be much easier than heavily relying on the reference genome as a template and mining these short read mappings themselves. In the remainder of this talk, I will first talk about our two frameworks. One of them would be a high-resolution nucleotide level alignment framework. The other one would try to make things a bit faster by reducing the resolution a bit in a fragment chaining framework. And finally, I will describe some practical considerations that we thought about for implementing Dissect and give you some experimental results from our tool. Our nucleotide level splice alignment framework could be described as follows. Imagine that we are given a transcript sequence of size n and also genomic sequence of size m, and also accompanying complement genome sequence that represents the complement characters for each of the bases here. And in this case, we will define an alignment from T to G with structural alterations as a function from the indices in T to the indices in G, G prime, and also a special gap character that represents that this particular transcript base was a single base insertion. And the score function here would be defined as the summation of base pairing scores. And depending on what kind of base pairing that is, it could have a different value for match, mismatch, or gap. And also, we will deduct the genomic penalty, transition penalty. And depending on what kind of transition there is between those two transcript bases, we might want to use a different type of penalty function. For example, we may want to further penalize backward transitions and inversion transitions rather than normal regular forward transitions. And for fusions, since we don't have a sense of the distance between the two sequences, we can only give a constant penalty value. And to further reward canonical introns, we can deduct some splice junction score from the overall genomic transition penalty to whenever there is some canonical splice on each end of the intron. To incorporate fusion cases, we have to introduce two more sequences. This would be S, the secondary genome sequence, and S prime, the complement of S. And in this case, we will extend the range of this function to the indices in S and S prime as well. And we have to introduce one fusion condition, which tells that if a certain transcript base is aligned to S or S prime, then every subsequent base has to as well. This guarantees that the fusion happens only once. A simple solution method for this could be described as follows. Imagine that we have an alignment matrix for aligning the transcript to the genome, and also another matrix for aligning the transcript to the complement of genome, in which case all of the forward transitions for the IJ cell would come from the right previous row and from all of the cells that come before in the genome. And all of the backward transition will come from the same row, but from every other cell that is after in the genome or at the same position. The inversion transitions will be taken from the complement alignment matrix, representing inverted junctions from the reverse strand alignment to the forward strand alignment. The final type of transitions we have for a single genome sequence would be for transcript insertions, in which case whenever we have a single base insertion at the ith base of the transcript, this would represent a single row transition from the previous row. And this would preserve the genomic position information from the previous aligned transcript base. For fusion transitions, we have to introduce two more alignment matrices. The first one is aligning the transcript to the secondary genome sequence, and the other one would be aligning the transcript to the complement of it. And in which case, we have quadratically many cells that we can take the fusion source to assign to this cell. Luckily, we don't have to consider this for all of the cells over here. We can pre-compute the optimal fusion sources and then compute them on the fly for computing these alignment matrices. And our complex is order of m squared with this simple algorithm for each row, and overall runtime complex would be order of nm squared, which is quite slow considering that m is actually the genome size. And thinking that this is a really general solution for all arbitrary gap penalties for even functions that looks like this, which we may have no practical use at all. And for most practical purposes, it will be much more useful to have a gap penalty function that looks more like this. And more than 20 years ago, Miriam Myers and Giancarlo independently showed that if the gap penalty function given to us satisfies this convexity property, then among these quadratically many transitions between ith row and i+1 row, we can actually process them in such an order that we can get away with not calculating most of them. And this would reduce the complexity of each row construction step from order of m squared to order of m log m, giving us an overall complexity of nm log m. And Giancarlo further noticed that this log m factor here actually comes from doing a binary search for solving this problem snippet. And if our gap penalty function is rather simple, like a logarithmic gap penalty function or a square root gap penalty function, then we can calculate x directly in constant time, in which case the overall complexity would reduce to order of nm, which is tight for aligned problems. For adapting this sparsification idea to our alignment model, we have to do some further work because our gap penalty function was PS minus JS, which is genomic transition penalty minus junction score, which may not necessarily be convex. Luckily, our JS function is rather simple, so we can split the entire set of forward transitions into two subcases. In the first case, we'll only consider the ones that correspond to canonical splice starting sites, and the second one will only consider the ones that do not correspond to canonical splice starting sites, and then after computing them separately, we will merge them by taking the maximum in each column. And this would give us the finalized I+1 row for forward transitions. And in order to handle the backward transitions, we can run the entire algorithm in a mirror image fashion from right to left and do all of the computations separately and merge them. And for inversions, we can simply take the entire ith row from the complement alignment matrix. And after we merge all of these 8 subcases into a single row, then we'd have, and do the computations for the entire alignment matrix, then we would have solved our transcript to genome alignment problem with structural alterations in order of nm log m time for convex gap penalty functions and nm time for simple convex gap penalty functions. This concludes the part of my presentation for our nucleotide level alignment framework. Now I will talk a bit about fragment chaining framework. The main difference between nucleotide level alignment and fragment chaining is that we are not given the transcript and the reference now as its nucleotides, but we are rather giving a set of fragments that represent homologous intervals on the transcript and the genome. In this case, if you think of this as a chain, then the transition from F1 to F2, the fragment 1 and fragment 2, would actually be a backward jump in the genome. And transition from F2 to F3 would be a inverted jump between the strands. And our first formulation upon this framework is a disjoint fragment chaining formulation, in which case we assume that each fragment has the following set of information. It has a score value, it has a transcript starting position, transcript ending position, genome starting and genome ending positions. And in this case, we define a valid chain of fragments as a sequence of fragments such that each fragment ends in a transcript before the next fragment starts. And we have a really simple score value definition for the score function definition for the chain. We first sum up individual fragment scores and then subtract fragment transition penalties for each adjacent fragment pairs, and then subtract the transcript insertion penalty for the beginning of the chain and also for the ending of the chain. And our penalty function could be defined identically to our nucleotide level alignment framework. We would have a penalty for transcript distance between the fragments, a genomic distance penalty depending on the distance and also the type of the transition, and also the junction score that we defined earlier. And as you may have noticed, this problem would actually reduce to our nucleotide level alignment framework if you consider each fragment as a single base. And a really simple solution method for this could be as follows. We would first initialize all of the fragment scores, and then for each of these fragments, we will identify the potential fragments that we can chain towards. And we would do this using our specialized transition penalty functions. Afterwards, we can finalize the fragment scores and then select the optimal chain of fragments and backtrack. And this is a very simple algorithm that works in k-square time for k fragments given. However, this may not be the most realistic formulation that we can come up with for fragment chaining problem, because in practice, if you have two exons head-to-head in a transcript, and if you are using ctDNA extend strategy for constructing these fragments, then you would tend to overextend if there is some homologous sequence between the exons. In which case, in order to relax our formulation for such instances, we redefined our set of constraints, and we said that throughout the chain, we require the transcript starting positions to monotonously increase and also transcript ending positions to monotonously increase. As you can see, this prevents further backwards jumps that are from further back from the beginning of the transcript, which will make the problem NP hard. So this problem is still in our polynomial alignment framework. The score function could be defined in the same way, with the only difference that the penalty right now would be calculated separately for disjoint pairs and overlapping pairs. And in our paper, we described several methods of computing this defining this overlapping pair penalty function and how to incorporate splice junctions into it. And now I would like to talk a bit about our alignment tool, Dissect, and give you some results from simulated data sets and also a real-life prostate cancer data set. Even though the fragment chaining formulation is much faster than our nucleotide level alignment formulation, in practice, it would still be infeasible to construct an entire set of fragments between the transcript and the whole genome. For that reason, the first step in Dissect would be to reduce the search space and try to identify a genomic subregion that is likely to contain the entire transcript, or a genomic region pair if the transcript is a fused transcript. And after that genomic region is inferred, then we would construct the fragment set using an extended version of our in-house alignment tool, Mrs. Fast, and then we will apply our fragment chaining solution. And in order to further improve the results, we will take the chain from the second step and further refine the gaps and overlaps using a nucleotide level alignment. And the finalized improved chain is our Dissect aligner's output. We first want to test Dissect for false positives, and for that we extract some RefSec mRNA sequences and align them directly with no modification, in which case we saw that more than 99.5% were actually aligned as wild type with no structural alterations at all. And when we increase the error rate to 1% with insertion, deletions, and substitutions, then we saw that there was a minor reduction in the amount of wild-type alignments to 99.4%. And when we increase it to 4%, which is higher than currently available Illumina machines, then we barely got under 99%. We also want to check whether Dissect is robust for novel insertions. And for that, we switched from mouse to human and extracted real human novel insertions from Kid et al. Study in 2010, and inserted these novel insertions to various exon breakpoints. And we saw that whenever the insertion length is more than 20 bases, we have a very little rate of false positive event detection. And whenever this insertion length is longer than 50 bases, we have about a bit more than 2% error rate detection. And then we want to test Dissect for false negatives. And for that, we simulated some artificial structure alteration events upon known gene annotation. And as you can see, among these different type of events, duplication, inversions, rearrangement, and fusions simulated, if you look at the diagonal, you can see that Dissect is actually quite specific to the type of the event that we simulate. It is highly unlikely that Dissect would actually report a different type of event when you simulate duplications, for example. And as you can see from this column, you can also see that Dissect is quite sensitive for most of the event types that we simulated. For most of these, this is more than 95%. There are some, for some cases, like short exon inversions, Dissect would lose about 10% of the simulated events. And the reason for that is the base pairing score that you would get from the exon might actually be less than the penalty that you would have on each side of the exon for the inverted transitions. And also for imbalanced fusions, Dissect actually lost quite a few of them. And the reason for that is not actually the alignment formulation, but it comes from the first genomic region inference step, because if you have a fused transcript and one side is really short, then it might make more sense to assume that that side is a novel insertion and assume that the entire transcript comes from the longer, and comes from the gene that contains the majority of the transcript. And we also tested Dissect in a real-life data set, and for that, we found some 50 base pair RNA-seq reads for prostate cancer cell line C42 and assembled them using Transcribed. And among a bit less than 600,000 contexts that we obtained, Dissect identified a bit more than 13,000 structural alteration events. And among these duplication, rearrangement, and inversion events that were reported, majority of them displayed more than 90% overlap with a single gene. And which means that if these are in fact real, then they might have some potential biological functions. And among the inversions that were reported, a significant majority of them were actual single breakpoint inversions, which is much more plausible from a biological perspective. And even though testing Dissect for specificity would require us to do lots of validation and would be really costly, we still want to check whether Dissect is sensitive enough. For that reason, we compared the fusion results with another fusion detection study by McPherson et al. With a tool called Comrade. And among the 8 gene fusion cases that were validated in that study, we did 2 types of tests. In the first test, we assembled the contigs and then aligned them with Dissect. And the second test, and this would actually represent what Dissect can do with currently available sequencing technology. In the second one, we want to see whether Dissect would actually would give good results for future sequencing technologies that could actually provide us with really long, accurate reads. And for that, we did not assemble them, but we directly extracted 200 base pair breakpoint sequences from the gene fusion breakpoints. And from the first case, in the first case with currently available technologies, Dissect was able to identify 6 of the fusion breakpoints correctly. And for the one that it was not reported, the assembler did not report any contig that spanned the breakpoint. And for the other one, the assembler reported a highly imbalanced fusion that Dissect wasn't able to predict correct gene location. The results are much better for the futuristic type of experiment, though. When we extract the breakpoint sequences ourselves, then Dissect was able to capture all of the breakpoint sequences correctly for all fusions. And since Dissect does not rely on any gene annotation, it did not actually report some of these as fusions, but one of them was reported as rearrangement and one of them was reported as wild type. And when we further compared this to a gene annotation, we could actually say these are in fact fusion breakpoints. And our study contributes 2 novel alignment frameworks. One of them is a nucleotide alignment, one of them is fragment chaining. And we also have an implemented tool now up on SourceForge, you can download it. And it has quite high sensitivity and specificity for most event types that we tested it for, with some exceptions over here. And also it is quite efficient. We were able to run the entire C42 data set, assembly data set, in less than a day using a single node. I would like to thank my former lab mates in SAP Lab of Computational Biology and also Transcribed development team in Genome Science Center for helpful discussions throughout this study, and also members of the MIT Berkeley Lab for helping me with this presentation. And these are our funding sources, and also I'd like to thank ISCB and NSF by a directorate for travel fellowship. And of course, I would like to thank to all of my co-authors, and also thank you for listening.